So good afternoon, everyone. My name is Bipasha, and thank you for attending my presentation on the research I'm involved with at the Shoyket Lab. The main goal of this project is to study the release of rod-derived co-imbiability factor from an injectable hydrogel drug delivery vehicle. Now, this is important for two main reasons. Rod-derived co-imbiability factor, or RDCVF, is a potential therapeutic against retinitis pigmentosa, or RP, which is the most common inheritable retinal degenerative disease affecting nearly 1.5 million people worldwide. And the second reason is that RDCVF is an extremely novel protein, and our group would be the first to show its incorporation and delivery from a drug delivery system. Now that we understand why this research is relevant, let's dive into some background about the project. And I'd like to draw your attention to the first text box in my poster. So retinitis pigmentosa is a genetic disease that affects the rod and cone cells of the eye that help us see. Rod cell degeneration leads to cone cell loss and eventually blindness. RDCVF is a recently discovered protein secreted by the rod cells that has been shown to maintain and increase cone cell viability and sensitivity. But there are major problems with delivering the protein therapeutics, especially to the eye, because topical applications are limited by rapid drug clearance and bolus injections by risk of overdose. So in order to mitigate these risks, we developed an affinity-based hydrogel drug delivery vehicle, which can sustain the release of RDCVF in the eye. I'd now like to draw your attention to this image here, where you can see our hydrogel solution that consists of an injectable physical blend of hyaluronan and methylcellulose, or HAMmock. The methylcellulose component of the gel is modified with a peptide binding partner of the SRC homology 3, or SH3 domain. This is important because we've also modified our RDCVF protein to express a SH3 domain, which interacts with its binding peptide partner on the hydrogel, and thus its release can be controlled. We ensured that the affinity between the peptide and the protein is weak so that sufficient protein can be released from the hydrogel. We hypothesized that our fusion protein retains bioactivity of RDCVF and that its release can be controlled from our modified hydrogel. So our research consists of three main steps, starting off with the expression and purification of RDCVF SH3 from E. coli. As you can see here, there's our fusion protein consisting of RDCVF, the SH3 domain, as well as His, flag, and strep tags. The His and strep tags are particularly important to the purification step of the protein. Once we've obtained purified protein, we assess its purity using SDS page, as seen here, and mass spectrometry, which confirm that the size of the protein obtained is within the expected range of 36 kilodaltons. This was also the step that I'm personally most involved in, creating large stocks of proteins for more studies. The second step is assessing the effectiveness of our novel protein using a bioactivity assay to ensure that the addition of the SH3 domain does not affect its functionality. With the help of our collaborators, we take chick retinal explants, we dissociate the cells, treat them with either RDCVF SH3 or a controlled PBS solution, and measure the viability of cone cells after 7 days. Here are some results we obtained from this assay, and I'd like to direct your attention to the first row of images where the green cells are all the viable cells. On performing a quantitative analysis of these green cells, you can see in graph B that the treated explants have a higher viability than the control PBS treatment. Now that we've ascertained that our fusion protein is bioactive, we can move on to the final and most crucial step of our research, which is studying the controlled release of RDCVF SH3 from our modified HAMmock hydrogel. To do so, we suspend the hydrogel in medium, let the protein diffuse out since it is an affinity-based system, and measure the amount of protein in the medium using a custom ELISA, which I can expand a bit more in the Q&A. As you can see in graph A, the cumulative protein released from HAMmock with peptide is much lesser than that of HAMmock alone, indicating some amount of control over the release of the protein. In figure B, we have plotted the fractional release of the protein with respect to the root of time. And as you can see here, the linear portion of this graph for hammock width peptide has been significantly greatly increased compared to the simple hammock gel. Using the slope of these graphs, we calculated the diffusivity of RDCBF SH3 from the hydrogels and inferred that the diffusivity is also significantly reduced with our modified hydrogel. And these three results show that the release of our protein can be controlled with our modified hydrogel. In conclusion, our group is one of the first to show purify and extract RDCBF SH3 to ensure its bioactivity as well as its controlled release from our modified hydrogel. Our future work includes testing the bioactivity of our released protein over time in vivo, which will bring us a step closer to a protein therapeutic against retinitis pigmentosa. Thank you very much. Stop sharing.